Welcome to rainy school days. Think back to those quiet afternoons in school when the world outside was grey and calm, rain pattering against the windows, the classroom a little dimmer, the pace a little slower. Lessons felt softer then, almost like stories told just for you. That's the spirit we keep here. In a world that shouts, scrolls, and overstimulates, this is the most peaceful version of school you can imagine. Together, we'll take our time, moving gently, slowly, and carefully through the fundamentals of knowledge. No rush, no pressure. Just the comfort of curiosity unfolding at its own pace, like reading a book by lamplight on a rainy day. And tonight, on this rainy school day, we'll be studying microbes, the tiny, hidden organisms that shape our bodies, our food, and our planet. We'll go slowly, step by step, so that even the most complex ideas feel clear and gentle. Think of it less like a science lecture and more like a quiet story time, where the smallest characters turn out to be the most important. A microbe is anything so small that you need help to see it. Some are living cells. Some are not really alive in the usual sense. All of them are tiny. Imagine a single strand of hair. If that hair were a highway, a typical bacterium would be a compact car parked sideways across one lane. A virus, even smaller, would be a pebble on the shoulder. These small things move the big things in life. In this session, we'll stay calm and curious. We'll keep explanations simple, and when a word sounds fancy, we'll slow down and unpack it. Our aim is depth without confusion. By the end, you'll have a sturdy mental map: what microbes are, how they live, how they help us, how they sometimes harm us, and why life on Earth would collapse without them. Take a breath. Get comfortable. Let your eyes soften. We'll go step by step. First, we'll meet the main characters. Bacteria are single-celled organisms with simple bodies but astonishing adaptability. Viruses are tiny packets of genetic instructions that must borrow a cell's machinery to make more of themselves. Fungi include yeasts and molds and the hidden networks beneath mushrooms. They recycle the world. Protozoa are single-celled, animal-like swimmers in drops of water and soil. Algae are plant-like, many of them microscopic, quietly filling the air with oxygen. We'll weave in stories, like how a curious craftsman polished lenses and first glimpsed animal cules, or how a splash of mold on a dish set medicine on a new path. We'll visit the immune system as if it were a city's defense team, and we'll look out over Earth's cycles, where microbes act like the planet's unseen maintenance crew. Let's begin. To understand microbes, start with size. If a human cell were a big house, a bacterium would be a small shed, and many viruses would be a doormat on the porch. At this scale, distance is different. A few micrometers to a bacterium feels like a stroll across town. Surfaces that look smooth to us are canyons and cliffs to them. Water isn't like our water. At microbial size, it's more like honey, sticky and resistant. This helps explain why tiny hairs and little tails are so useful to them. Microbes are everywhere. They ride dust in the air. They hide in the folds of leaves and the cracks of rocks. They live in boiling springs and in sea ice, in deep caves and in your gut. Most are harmless. Many are helpful. A small fraction can cause disease. If you imagine a stadium full of people, the fraction who cause trouble would be a handful in the upper deck. The rest are just living their microscopic lives. How did we find them? For most of human history, we didn't know they existed. In the 1700s, a self-taught lens grinder named Antony van Leeuwenhoek polished tiny glass lenses until he could see things no one had seen before. He peered into drops of water and wrote about little animals darting about. Later, Louis Pasteur showed that microbes sour wine and spoil broth, and that heat could tame them. Joseph Lister applied the idea to surgery, cleaning instruments, and saving lives. Robert Koch connected specific microbes to specific diseases, showing that invisible causes can have visible, predictable effects. Then, in the early 20th century, Alexander Fleming noticed that mold on a dish had killed nearby bacteria, and the first widely used antibiotic, penicillin, began its journey. We tell these stories not as trivia, but as reminders. Curiosity plus simple tools can open entire worlds. That is the spirit we'll keep tonight. Patient, precise, unafraid of small details, and always returning to the big picture. Let's meet our first major group in depth. Bacteria are single-celled organisms. Each cell is a self-contained life. Picture a tiny water balloon wrapped in a tough jacket. The flexible inner membrane is the balloon. The tough jacket is the cell wall, which keeps the cell from bursting and gives it shape. Inside, instead of a separate room for DNA, the genetic material sits in an open space, coiled like a ribbon in a backpack. Scattered around are ribosomes, the cell's little factories for making proteins. Some bacteria have one or more whip-like tails called flagella. These tails spin like propellers, helping the cell swim through what feels, at that scale, like syrup. Different bacteria wear different jackets. Some walls are thick and layered. Some have a thin wall plus an outer covering. In the lab, a dye test called gram staining helps tell these types apart. You don't need the chemistry tonight, just the idea that wall design matters. It affects how a bacterium lives, how it protects itself, and how doctors choose medicines when infections happen. Shapes are simple and helpful for a first pass. Some bacteria are round like tiny beads. Others are rod-shaped like little hot dogs. A few are corkscrew spirals. If you've ever looked at pepper under a microscope, you've seen how simple shapes can create many patterns. Bacteria arrange themselves in chains, clusters, and pairs, like beads on strings or grapes on a stem. How do they make more of themselves? They usually grow and split. A cell gets larger, copies its DNA, and divides into two. Under ideal conditions, that division can happen quickly. In less than the time it takes to watch a movie, one cell can become a crowd. Of course, real life isn't ideal. Food runs out, waste builds up, temperatures change, growth rises, stalls, and falls again. You can imagine a small town that expands when jobs are plenty and slows when resources thin. Bacteria are adaptable. They can trade small loops of DNA called plasmids, little bonus rings that may carry useful tricks. If one bacterium learns a new way to survive, it may pass that idea along. They can also pick up stray pieces of DNA from their environment or be visited by viruses that accidentally move genes from one cell to another. If this sounds like a bustling marketplace of ideas, that's a good picture. Bacteria are constantly sharing, testing, and refining survival strategies. Some bacteria build protective shells called endospores. Imagine a seed with its vital parts sealed away. In harsh conditions like heat, drying, or chemicals, the cell packages its essentials and waits. When conditions improve, the seed wakes up. This is not common to all bacteria, but where it exists, it makes survival across time and distance possible. Let's talk about metabolism in everyday terms. Metabolism is just how a cell gets energy and materials. Some bacteria breathe oxygen like we do. Others prefer to live without it. Some munch on sugars and starches. Others feed on unusual things, like sulfur, iron, or even oil. Near ocean vents, where hot, mineral-rich water gushes out, bacteria harvest energy from chemicals the way plants harvest energy from sunlight. They are Earth's ultimate improvisers. What do bacteria do for us? Quietly. Almost everything. They decompose fallen leaves and dead trees, returning nutrients to the soil. They help plants grow by making nitrogen usable. In our intestines, friendly bacteria turn tough fibers into helpful compounds, make vitamins, and crowd out invaders. In food, they turn milk into yogurt and cheese, cabbage into sauerkraut and kimchi, soybeans into miso and tempeh, and cocoa beans into chocolate with depth and aroma. In industry, we ask engineered bacteria to make medicines like insulin, to build enzymes that help detergents clean at lower temperatures, and to produce bio-based materials. Can bacteria cause disease? A small set can. They may enter through a cut, ride in water or food, or spread through the air. When harmful bacteria find a hospitable place, they may release toxins, invade tissues, or trigger strong immune reactions. Strep throat, some forms of pneumonia, tuberculosis, and certain food poisonings are bacterial. The good news is that we have antibiotics, medicines that target bacterial processes. The caution is that overuse and misuse of these drugs have encouraged some bacteria to evolve resistance. Think of it like weeds in a garden. If we use the same tool too often, the hardiest weeds remain and slowly take over. Stewardship, using the right drug only when needed and finishing the course, helps preserve these tools. Bacteria often live together in communities called biofilms. Picture a slimy layer on a rock in a stream, or the plaque on your teeth in the morning. In a biofilm, cells stick to a surface and to each other, sharing nutrients and sending chemical messages. Like a small town with shared roads and services, biofilms are more resilient than single wandering cells. This makes them hardier in nature, and sometimes harder to eliminate in medical settings. If you hold one image of bacteria, make it this: small, simple, social survivors. They are chefs, recyclers, engineers, and, occasionally, troublemakers. Without them, the world would stall. Viruses are different. A bacterium is a complete cell. A virus is a packet of instructions wrapped in a protective coat. On its own, a virus is quiet. It does not eat, grow, or make energy. But when it meets the right cell, it becomes a burglar with a very specific lockpick. A typical virus carries genetic material, either DNA or RNA, tucked inside a protein shell called a capsid. Some have an extra outer layer, a kind of fatty envelope borrowed from the last cell they left. The shapes are beautiful in their simplicity. Icosahedrons that look like tiny soccer balls, long rods, or complex lunar lander shapes in the case of bacteriophages, the viruses that infect bacteria. Every virus has a preferred door, called a receptor, that it recognizes on a host cell. When the match is right, the virus attaches and either injects its genetic instructions or enters the cell whole. Inside, it redirects the cell's factories. Instead of building the cell's own parts, the hijacked factories begin to assemble copies of the virus. Eventually, those copies leave, sometimes bursting the cell, sometimes sneaking out wrapped in a bit of the cell's membrane. There are two broad rhythms to this process. In a fast, lytic rhythm, the virus enters, makes many copies quickly, and breaks the cell open to release them. In a quieter, lysogenic rhythm, the viral instructions can lie low, tucked into the host's DNA, copied along with the cell for a while, and then wake up later. Think of the difference between a sudden storm and a long grey drizzle that builds to rain. Are viruses alive? It depends on your definition. If alive means doing life's work on your own, then no. If alive means capable of evolution, adaptation, and reproduction when given a host, then they sit on the threshold. Either way, they have shaped life profoundly. Viral infections push immune systems to sharpen their tools. Genes have sometimes jumped from viruses into hosts, leaving permanent marks in DNA. In the oceans, viruses constantly prune and balance microbial populations, recycling nutrients in the process. Some viruses cause mild illness, others can be severe. Colds, flu, measles, chickenpox, and many stomach bugs are viral. Treatments differ from bacterial infections because antibiotics don't work on viruses. Antiviral drugs target specific steps in the viral life cycle, and vaccines teach the immune system to recognize key features. A vaccine is like a wanted poster. If you see this shape, respond fast. Once the immune system has that memory, future encounters go better. There is a hopeful twist to the virus story. Bacteriophages, the viruses that infect bacteria, can be allies. Phages are everywhere. Bacteria are. Soil, water, our bodies. Because some phages are very specific about which bacteria they attack, they have been explored as a way to control bacterial infections, especially when antibiotics fail. The field is careful and growing, but the idea is elegant. Use a predator nature already employs, with narrow focus and built-in amplification. When you think virus, you might think danger. Replace that with a more complete picture. A tiny genetic messenger that must borrow a cell to make more of itself. Sometimes that borrowing harms us. Sometimes it barely registers. Always, it nudges evolution forward and reminds us how intertwined all life is. Let's widen the circle and meet 3 more groups that complete our beginner's map: fungi, protozoa, and algae. Each deserves its own course, but tonight we'll build clear, sturdy sketches. Start with fungi. If bacteria are solo artists and viruses are burglars, fungi are the recyclers and network builders. Many live as long, branching threads called hyphae. Together, those threads form a mycelium, a hidden net that winds through soil and wood. When conditions are right, some fungi send up fruiting bodies we recognize as mushrooms. Others stay microscopic. Yeasts are single-celled fungi that breathe sugar and release carbon dioxide and alcohol. That simple habit lifts loaves and ferments beer and wine. Fungi are essential because they take apart tough materials like wood and leaf litter. They unlock nutrients and hand them back to the world. If you've ever watched a fallen tree soften and return to the forest floor, you've watched fungal patience in action. Some fungi live in partnership with plant roots, forming mycorrhizae, networks that help plants pull in water and minerals. In return, the plant shares sugars it makes from sunlight. It is a trade agreement older than forests. Fungi can also cause trouble. Athlete's foot and some yeast infections are fungal. Certain molds produce toxins in spoiled foods. In people with weakened defenses, fungi that are usually harmless can invade more deeply. But remember the balance, the recycling, the partnerships, the bread and cheese and medicine. Penicillin began with a mold. Protozoa come next. These are single-celled, animal-like organisms that move through water films in soil and ponds, or along the lining of a gut. Some swim with tiny hairs like oars. Some pull themselves along with whip-like flagella. Others ooze with small, shape-shifting feet. Under a microscope, they look lively, turning, gulping, splitting. Many are predators of bacteria, keeping microbial communities in balance. A few cause disease. The agent of malaria is a protozoan that rides in the bite of a mosquito and takes up residence in human blood cells and liver cells, cycling through stages like an actor changing costumes between acts. Finally, algae. Many algae are microscopic and drift with currents in lakes and oceans. These are the phytoplankton, small photosynthetic factories that use light to fix carbon and release oxygen. step outside and take a breath. A significant share of that oxygen was made by algae. You don't see them, but they are the lower floors of the marine food web, feeding tiny animals that feed bigger ones, up to fish and whales. Larger algae, the seaweeds, are also important, providing habitat and food along coasts. Sometimes algae bloom too richly when waters get overloaded with nutrients, and that can harm ecosystems. But in a healthy balance, algae are planet-shaping. If you hold one sentence for this section, let it be this: fungi return what's fallen, protozoa keep the mix lively, and algae quietly fuel the planet with light. Now let's come home to the human body. We are not a single species walking around alone. We are a cooperative apartment complex. Trillions of microbes live in and on us, most peacefully, many helpfully. Together they are called the microbiome. The richest community sits in the gut. Imagine a crowded marketplace, stalls lining the street, vendors trading fiber for helpful molecules, and neighbors chatting constantly. Our own enzymes can't fully break down many plant fibers. Gut microbes finish the job and pay us in small molecules that nourish our colon and influence metabolism and inflammation. They synthesize certain vitamins. They train the immune system, especially early in life, teaching it when to react and when to relax. A diverse, balanced microbial community is like a balanced neighborhood, a mix of residents that keeps the peace. The skin has its own blend, microbes that like the drier forearm, the oilier nose, the humid nook behind the knee. They form a living shield. They take up space and resources, so invaders have a harder time settling in. The mouth hosts vibrant communities too. Daily brushing and flossing aren't about sterilizing. They're about guiding the community away from plaque and inflammation toward a healthier mix. Sometimes the balance tips. A course of antibiotics, while life-saving when needed, can scramble the gut community for a while. Diet shifts can shift microbes. Illness can do it too. When the mix changes in unhelpful ways, we call it dysbiosis, literally bad living together. Scientists are exploring how this links to digestive troubles and beyond. While the field is still evolving, a common-sense picture has emerged. Varied whole foods tend to support a more resilient microbial neighborhood than a narrow, highly processed diet. Now, pathogens, the microbes that cause disease, also interact with us. Some bacteria make toxins, some viruses hijack cells, some protozoa invade tissues. We respond with an intricate defense, the immune system. Think of it as a city's layered security. The skin and mucous membranes are walls and moats. Stomach acid is a chemical barrier. Inside, roaming cells patrol like officers, sensing patterns that mark invaders. They eat what they find or call for backup. Deeper still, specialized cells learn the unique signatures of particular microbes and remember them. If that microbe returns, the response is faster and sharper. This is immunity. Vaccines make use of that memory. They present a safe look-alike, a fragment, a weakened form, a carefully designed mimic, so the immune system can practice. Think of it as a training exercise with realistic props. If the real intruder arrives later, the guards recognize the face and act swiftly. Across history, this practice has turned deadly childhood illnesses into rare stories and has protected communities when enough people join in. Antibiotics were another revolution. These medicines target bacterial features, like making cell walls or copying DNA, in ways human cells don't use. Used correctly, they save lives every day. But because bacteria evolve quickly, the more we rely on a drug, the more we select for survivors that can dodge it. Resistance is not a moral failing of bacteria, it's evolution at work. Our task is to use antibiotics wisely, only when needed, the right choice for the right time, guided by testing whenever possible. Microbes also star in our kitchens. Yeasts make bread rise and grapes transform. Bacteria turn milk into foods that keep longer and taste deeper. Fermented vegetables develop complex tang. These foods aren't just tasty, they're cultural, handed down, shared, and quietly shaped by local microbes. When you bite into a sourdough crust, you're tasting a community's history. Medicine, too, relies on microbes beyond antibiotics. Many vaccines are grown with their help. Insulin, a life-saving hormone for diabetes, is now produced by bacteria taught to make it. Enzymes used to diagnose and treat are often microbial. Even tools that let scientists edit genes, like CRISPR, were discovered as part of bacterial immune strategies against viral invaders. The relationship between humans and microbes is not a battle with winners and losers. It is a long negotiation, with daily trade, occasional conflict, and constant learning on both sides. When we support balance, through hygiene that's smart but not harsh, through food that nourishes us and our microbes, through medicine that's careful and targeted, we live well together. Step back from the body and picture Earth as a living system. Microbes are the maintenance crew, the recyclers, the engineers, and the emergency responders. Consider the nitrogen cycle. Nitrogen is a basic ingredient in DNA and proteins, but the form floating in the air is like money locked in a safe. Certain bacteria have the combination. They take nitrogen gas and convert it into forms plants can use. Some of these bacteria live freely in soil, others live in nodules on plant roots, especially beans and peas, exchanging fertilizer for sugars. Later, different bacteria return nitrogen to the air, closing the loop. Without these conversions, fields would starve and forests would fade. The carbon cycle also depends on microbes. When leaves fall and animals die, bacteria and fungi take them apart. Carbon atoms travel back into the atmosphere as carbon dioxide or are carried deeper into soil. In the oceans, microscopic algae fix carbon with sunlight, feeding the marine food web and helping move carbon into deeper waters when they die and sink. Viruses in the sea help break cells open, recycling nutrients in place and influencing how much carbon is locked away. Microbes shape climate. Some produce methane as they digest in oxygen-poor places like wetlands or the bellies of cows. Others consume methane. Some produce nitrous oxide. Some pull carbon dioxide from the air. These tiny processes, multiplied by unimaginable numbers, add up to significant flows of greenhouse gases. Understanding microbial communities is now part of understanding climate change and its solutions. They also form remarkable partnerships. Lichens, those crusty patches on rocks and trees, are alliances between fungi and algae or cyanobacteria. The fungus builds a house. The partner does photosynthesis and pays the rent in sugars. Mycorrhizal fungi form networks between tree roots that can shuttle water and minerals, and perhaps even signals, the so-called wood-wide web. Coral reefs rely on algae living inside coral animals. When that partnership breaks under stress, corals bleach. Microbial partnerships are everywhere once you know to look. When humans cause messes, microbes often help clean them. In wastewater treatment, bacterial communities break down organic matter and help remove nutrients before water returns to rivers. After oil spills, certain bacteria feed on hydrocarbons and help degrade the mess. This is called bioremediation, guiding nature's cleaners to do the work they already know how to do. In mines and polluted soils, specialized microbes transform toxic compounds into safer forms. Through careful, contained engineering, We can even teach microbes to capture carbon or make biofuels, aligning industrial processes with biological cycles. If you imagine Earth as a vast, breathing organism, microbes are its red blood cells, its liver, and its gardeners. They move elements where they're needed, filter waste, and tend the living spaces that make the rest of life possible. We've covered a lot of ground. Let's stitch it together with a slow, simple recap. Microbes are tiny, but they sit at the center of life. Bacteria are single cells with simple parts, adaptable and social, sometimes helpful, sometimes harmful. Viruses are genetic travelers that need a host cell to copy themselves. They challenge us, but they also drive evolution and balance ecosystems. Fungi pull the world apart so it can be put together again, partnering with roots and baking our bread. Protozoa are lively single cells that graze and hunt in water films and sometimes cause disease. Algae, many of them microscopic, capture light and feed the ocean, gifting us oxygen with every sunrise. Inside us, microbes form communities that help digest food, tune the immune system, and protect our skin and mouths. When pathogens invade, barriers and immune cells stand guard, and memory from past encounters, including vaccines, helps us respond fast. Antibiotics are powerful tools for bacterial infections, and careful use keeps them powerful. In the kitchen, microbes create flavors, preserve foods, and connect us to tradition. In medicine and industry, they manufacture, diagnose, and inspire. Across the planet, microbes run the cycles of nitrogen and carbon, shape climate, build partnerships with plants and animals, and clean up messes. Through them, matter is recycled and life stays possible. If a single sentence must hold the course, let it be. Microbes are the invisible infrastructure of life. When we talk about microbes, it's easy to forget that for most of human history, people had no idea they existed. Illness was blamed on spirits, bad air, or curses. Food spoiled mysteriously. Wine turned sour. Bread failed to rise. It wasn't until people found ways to see microbes that this invisible world became real. So let's spend some time walking through the main ways scientists study microbes. The first step was the microscope. Imagine holding a tiny droplet of pond water under a curved piece of glass. To the naked eye, it's just water. But when Antony van Leeuwenhoek polished his handmade lenses in the late 1600s, he saw animalcules, little animals, swimming and darting about. His instruments were simple, but they opened a new universe. Modern light microscopes use carefully arranged lenses and beams of light. They magnify not just once, but in stages, making it possible to see bacteria and some larger structures inside cells. Special techniques like phase contrast or dark field microscopy bend light in clever ways, highlighting details that would otherwise stay hidden. It's like adjusting a flashlight to reveal scratches on a surface you thought was smooth. But light has limits. Objects smaller than about 200 nanometers blur together because that's the wavelength of visible light. To go smaller, scientists turned to electron microscopes. Instead of light, these machines use streams of electrons, which have much shorter wavelengths. When electrons bounce off a sample, they can be collected and turned into images with astonishing resolution. Electron microscopes can reveal the ridges and grooves on the surface of a virus, or the layered structure of a bacterial cell wall. The images are black and white at first, often later colorized to help our eyes. Looking at them is like staring into an alien landscape. Seeing isn't the only challenge. Most microbes are nearly transparent. This is where stains come in. By adding dyes that bind to certain parts of a cell, scientists create contrast. The famous Gram stain invented in the late 1800s, uses a sequence of dyes and washes to distinguish between bacteria with thick walls and those with thinner walls. Gram-positive bacteria stain purple, while gram-negative bacteria turn pink. With a glance under the microscope, a scientist can gather a first clue about what they're dealing with. Another approach is culturing. This means giving microbes the right environment to grow. On a dish of nutrient-rich gel called agar, colonies of bacteria appear like small dots, each dot a clone of a single ancestor. Different species may produce colonies with unique colors, shapes, or textures. Culturing allows scientists to isolate microbes, study their behavior, and test how they respond to different conditions. It's like inviting seeds to sprout in a garden so you can observe their flowers. But not all microbes are easy to grow. Many are picky, requiring exact nutrients or environments we don't yet know how to reproduce. This is where molecular techniques step in. By extracting DNA from a sample, say, a handful of soil or a swab from skin, scientists can read the genetic code of the microbes present, even if they refuse to grow in the lab. This is called sequencing. Instead of seeing the organisms directly, we read their instruction manuals. Imagine walking into a library, picking up dozens of anonymous books, and sorting them into families based on language and script. That's what sequencing does. It tells us who's there and what they might be capable of. Together, these tools, microscopes, stains, culture, sequencing, form the eyes and ears of microbiology. They turn the invisible into the visible, the mysterious into the measurable. Every time we adjust a lens, color a cell, or decode a strand of DNA, we're peeling back another layer of the unseen world. When we imagine bacteria, we often picture them as lone swimmers, each cell drifting in liquid like a fish in the sea. But bacteria, like humans, are social. They often prefer to live together in communities called biofilms. To understand a biofilm, it helps to think of a city. The first step in building a city is settlement. A few bacteria land on a surface, maybe a rock in a stream, the inside of a pipe, or the enamel of a tooth. If conditions are right, they stick. They begin producing a sticky substance, like glue, that anchors them in place. This slime, called extracellular polymeric substance, is the mortar that holds the city together. As more cells arrive, they join the growing community. They arrange themselves in layers, forming towers and channels. The channels allow water and nutrients to flow in, and waste to flow out. It's remarkably similar to streets and sewers in a city. Inside, bacteria communicate with each other using chemical signals. This quorum sensing lets them coordinate. It's as if they are sending text messages. We've reached a critical mass. Time to build defenses. In this community, different roles emerge. Some cells keep dividing. Others go dormant, like night shift workers resting until needed. Some produce substances that protect against antibiotics or immune attacks. Together, they are stronger than they would be alone. We can see biofilms in everyday life. That slimy layer on a river stone, a biofilm. The slippery coating inside plumbing pipes, another biofilm. The dental plaque that builds up overnight on your teeth, a biofilm, complete with food webs and communication networks. Biofilms can be helpful. In wastewater treatment plants, engineered biofilms break down pollutants and clean water. In nature, they recycle nutrients and provide food for small organisms. But in medicine, biofilms can be a problem. When bacteria form biofilms on catheters, artificial joints, or heart valves, they become difficult to remove. Antibiotics have trouble penetrating the slime. The immune system struggles to reach them. It's like trying to eliminate crime in a fortified city, rather than chasing a single pickpocket. By studying biofilms, scientists learn not just about bacteria, but about cooperation and survival strategies. These communities remind us that even the smallest organisms find strength in numbers, building miniature civilizations we can only glimpse under a microscope. Antibiotics changed the world. Before their discovery, even small wounds could turn deadly. With antibiotics, doctors gained a powerful tool to kill bacteria and save lives. But bacteria are not passive targets. They adapt, they evolve, and this has led to the growing challenge of antibiotic resistance. To understand it, picture a garden full of weeds. You spray a weed killer, and most of the weeds wither. But a few happen to survive. Maybe their roots run deeper, or their leaves repel the spray. Those survivors grow and spread. Soon the garden is full of weeds that the spray no longer harms. The same thing happens with bacteria. When we use an antibiotic, most bacteria die. But sometimes, a few have a genetic quirk that lets them resist. Maybe their cell wall keeps the drug out. Maybe they produce an enzyme that breaks the drug down. Maybe they pump the antibiotic back out before it can do damage. Whatever the trick, it gives them an advantage. They survive, multiply, and pass the resistance on. Over time, the once powerful drug loses its effect. Resistance spreads not just vertically, from parent to child, but also horizontally. Bacteria can share resistance genes through plasmids, little rings of DNA they swap like trading cards. A resistant bacterium can pass its secret to neighbors, even across species. This makes resistance spread quickly in hospitals, farms, and communities. The problem is worsened by overuse and misuse. Taking antibiotics for viral illnesses, like colds, doesn't help, but still applies pressure on bacteria in the body. Not finishing a prescription leaves survivors behind. Using antibiotics in livestock feed to promote growth exposes entire populations of bacteria to low doses, encouraging resistance. What's the solution? Careful stewardship. Doctors now emphasize prescribing antibiotics only when needed and choosing the right drug for the right infection. Researchers are developing new antibiotics, but it's a race with evolution. Alternatives like phage therapy, vaccines, and improved hygiene can also reduce the burden. It's important to see resistance not as villainy, but as evolution in action. Bacteria are simply doing what life does, adapting to survive. Our task is to be wise stewards of the tools we have, to preserve their power for as long as possible. When people first learned that microbes cause disease, the reaction was understandable. Eliminate them all. Sterilize. Disinfect. Scrub until everything shines. But the truth is more nuanced. Not all microbes are bad. Many are helpful. Our immune systems even seem to need regular exposure to microbes to stay balanced. This is why we talk about finding the hygiene sweet spot. Think of the immune system as a security guard. If it never sees any challenges, it may overreact when something small comes along, like pollen or dust, leading to allergies or asthma. On the other hand, if it is constantly overwhelmed by threats, it becomes exhausted and strained. The goal is training, enough exposure to real challenges to stay sharp, but not so much that it becomes dangerous. Daily hygiene practices help. Washing hands with ordinary soap physically removes microbes, like rinsing pebbles from your hands after gardening. Soap doesn't need to kill everything. It just helps wash them away. Disinfectants are stronger tools, but they are best reserved for special situations like cleaning after illness or sanitizing hospital surfaces. Using them everywhere, all the time, can disrupt healthy microbial communities without real benefit. Children who grow up spending time outdoors, playing in soil, or living with pets often encounter a wider variety of microbes. Studies suggest this diversity of exposure may lower the risk of allergies and autoimmune disorders. It's as if the immune system, trained on many harmless visitors, learns to distinguish friend from foe more gracefully. The hygiene sweet spot is not about living in dirt or living in sterility. It is about balance. Keep hands clean before eating. Prepare food safely. Cover wounds. But also don't fear the ordinary microbes of everyday life. A walk in the forest, a garden under your fingernails, or the simple act of breathing fresh air. All of these are gentle teachers for the immune system. So when you think about hygiene, remember it's not a war of total elimination. It's a balance between cleanliness and connection, between protecting ourselves from harm and allowing our bodies to learn from the microbial world we live in. If you think of life as a library, genetics is the catalogue that tells each book how to write itself. In microbes, this catalogue is compact, portable, and surprisingly social. Bacterial DNA is usually a single circular chromosome, like a belt with no buckle, coiled tightly inside the cell. Many bacteria also keep plasmids, small rings of DNA that act like bonus booklets. A plasmid might carry instructions for digesting a rare sugar, tolerating a metal, or surviving an antibiotic. Plasmids are handy because they can be shared. Microbes don't just pass genes from parent to child, they pass them sideways, from neighbor to neighbor. This is called horizontal gene transfer. There are 3 main ways to do it, and each has a personality. The first is transformation. A cell picks up free DNA from its environment, shreds and scraps left behind when other cells burst or die, and tucks it into its own genome. Imagine walking past a bulletin board, spotting a useful recipe, and copying it into your notebook. Some bacteria are naturally competent. They're always ready to take notes. The second is conjugation. 2 bacterial cells connect using a tiny tube, often called a sex pilus, and one cell passes a plasmid to the other. This looks like lending a neighbor your favorite cookbook. Conjugation can bridge species boundaries, which is one reason useful traits, like antibiotic resistance, can spread through mixed communities. The third is transduction. A virus that infects bacteria, called a bacteriophage, makes a mistake while packaging DNA and carries a fragment of one bacterium's genes into another. It's like a courier delivering the wrong mail, but accidentally sharing a brilliant idea. Inside the cell, DNA is read and turned into action through transcription and translation. In transcription, the cell copies a bit of DNA into RNA, a temporary note. In translation, tiny factories called ribosomes read that RNA and build a protein, one amino acid at a time. If DNA is a recipe, RNA is the handwritten card you set beside the stove, and proteins are the meal. Microbes regulate this flow carefully. When sugar is scarce, a bacterium saves energy by not producing enzymes it doesn't need. When sugar appears, it flips genetic switches and begins cooking immediately. This on-off control is done with proteins that bind DNA like paperclips, turning pages the cell should read and hiding the rest. In changing environments, fast regulation is survival. Viruses bend genetics to their will. Some carry RNA instead of DNA and must copy it to make more of themselves. Retroviruses reverse the usual flow, turning RNA into DNA and stitching it into the host's genome. Once stitched, that viral DNA can sit for a long time, like a bookmark left by a previous reader. Genetic mistakes, mutations, happen naturally. Sometimes a letter is miscopied. Sometimes a chunk is inserted or deleted. Most changes are neutral. A few are harmful. A few offer an advantage. In huge populations that divide quickly, These small changes are a powerful engine of evolution. Give bacteria a new challenge, and some member of the crowd will stumble onto a solution. If it helps, that solution spreads. Modern microbiology reads and edits these texts. Sequencing lets us identify microbes by their genetic fingerprints. CRISPR, borrowed from bacterial immune systems, acts like a guided pair of scissors, allowing scientists to cut DNA at chosen spots. With careful rules and strong ethics, these tools promise new medicines, cleaner manufacturing, and crops that need fewer chemicals. They also demand humility. When you can rewrite recipes, you must be sure you understand the kitchen. At heart, microbial genetics teaches a simple lesson. Information is life's most portable tool. Microbes copy, share, and edit that information with incredible agility, letting tiny cells keep pace with a big, changing world. Metabolism is just a fancy word for how cells make a living, how they break food into energy and use that energy to build themselves. In microbes, metabolism is diverse and creative. If life were a city, microbes would run every restaurant, from vegan cafes to barbecue joints to experimental kitchens that turn rocks into dinner. Think first about energy. Many microbes extract energy by moving electrons from one molecule to another, like tapping a tiny battery. In oxygen-rich places, bacteria use oxygen as the final electron acceptor, like we do, because it's efficient. In oxygen-poor places, they improvise. Some use nitrate, some use sulfate, some use iron. Down in lake mud or in the rumen of a cow, whole communities are arranged by who can breathe what. It's a layered theatre of chemistry. Then there are the photosynthetic microbes, algae and cyanobacteria that use light to power their work. With sunlight, they fix carbon dioxide into sugars and release oxygen as a byproduct. Every sunrise is a factory whistle for these cells. Their work underpins food webs on land and across the oceans. Others practice fermentation, which is like extracting energy in a dim room when you can't find the light switch. Yeasts ferment sugars into alcohol and carbon dioxide. Lactic acid bacteria turn sugars into lactic acid. Fermentation yields less energy per bite, but it's quick and requires no oxygen. In a sealed jar of cabbage, lactic acid bacteria outcompete others by souring the neighborhood. In dough, yeast inflate gluten with bubbles until heat sets the structure. Growth follows energy. In a fresh broth or a new wound, bacteria often show logistic growth, a sleepy lag phase while they unpack their tools, a busy log phase where they divide rapidly, a stationary phase when food runs low and waste builds up, and eventually a decline phase. This rhythm is so reliable that microbiologists use it like a clock to time experiments. Temperature, salt, pH, and pressure all shape growth. Some microbes love heat, thermophiles, bathing happily in hot springs. Others prefer the cold, psychrophiles, thriving in sea ice. Halophiles like salt flats. Acidophiles enjoy sour mines. Barophiles withstand deep-sea pressures that would crush steel. Each microbe is tuned like an instrument for its chosen concert hall. To survive dry spells or harsh chemicals, some bacteria form endospores, the survival capsules we mentioned earlier. Others switch to cooperative living in biofilms, sharing nutrients and protection. Many alter their metabolism when conditions shift, like a restaurant changing menus with the seasons. For humans, microbial metabolism is opportunity. In bioreactors, we feed microbes sugars or gases and ask them to brew products: insulin, vitamins, enzymes, biodegradable plastics, even fuels. We borrow their pathways, trimming and tuning them with genetics to make cleaner processes. The big picture is simple. Microbes eat what's available, breathe what the environment offers, and grow as fast as conditions allow. Their creativity in doing so makes Earth flexible, capable of supporting life in places we once thought impossible. Imagine your body as a walled city. The innate immune system is the outer guard, always on duty, quick to act, not picky about details. The adaptive immune system is the detective squad, slower to start, but precise and blessed with memory. The first line is physical: skin, like a castle wall; mucus, a sticky moat; cilia in the airways, tiny brooms sweeping debris; stomach acid, a chemical furnace. When microbes cross these barriers, sentry cells, macrophages, neutrophils, dendritic cells rush in. They recognize patterns common to invaders: bits of bacterial walls, viral RNA, fungal sugars. Recognition triggers inflammation. Blood vessels open. Signals call reinforcements. Heat rises slightly. It's uncomfortable, but it's the city ringing the alarm. Macrophages and neutrophils engulf invaders, digesting them in enzyme-filled sacs. Dendritic cells do something special. They collect fragments and travel to the lymph nodes, our city halls, where they present these fragments to the adaptive system. There, T cells and B cells wait with receptors shaped by chance. Each cell's receptor fits a different molecular key. When a key fits, that cell activates and begins to multiply. Helper T cells coordinate the response, releasing signals that guide the force. Cytotoxic T cells patrol for infected host cells, showing viral fragments and order them to self-destruct. B cells turn into plasma cells that secrete antibodies, Y-shaped proteins that bind invaders, marking them for cleanup or blocking them from attaching to our cells. Antibodies come in classes with different jobs. Some patrol the blood, others line mucous membranes. Some cross the placenta to protect newborns. After the battle, most activated cells die back, but a subset becomes memory cells, lingering for years or decades. If the same microbe returns, these veterans respond fast. Vaccines practice this dance safely. By showing the immune system a harmless version or piece of a microbe, or by delivering instructions for our own cells to briefly display that piece, vaccines build memory without the risks of severe illness. When enough people are immune, the microbe struggles to spread. This is community protection, often called herd immunity. It protects those who can't be vaccinated and slows outbreaks. The immune system must walk a tightrope. Too weak, and infections flourish. Too aggressive or misdirected, and we suffer allergies or autoimmune disease. The microbiome helps train balance, teaching the system daily what a non-threat looks like. In simple words, immunity is recognition and memory. It knows friend from foe, remembers past battles, and keeps a logbook so the next encounter is shorter and kinder. Let's zoom out from cells to communities. Public health is what we do together to keep everyone safer. Clean water, sanitation, food safety, education, surveillance, and vaccination. Vaccines are the quiet champions here. A good vaccine chooses a target, a protein or structure the immune system can recognize, and presents it clearly. Some vaccines use inactivated or weakened microbes. Others use only a fragment. Newer ones use genetic instructions that let our own cells make the fragment for a short time. The result is similar. The immune system meets a wanted poster, takes notes, and files the memory. Public health also means watching patterns. When unusual clusters of illness appear, investigators trace contacts, test water or food, and act quickly, closing a source, issuing advisories, or organizing clinics. The famous story of John Snow in 19th century London is a classic. He mapped cholera cases and traced them to a contaminated water pump. When the handle was removed, cases declined. It wasn't magic, it was microbial logic. Trust is essential. Clear communication, transparency about risks and benefits, and respect for concerns help communities decide together. Most vaccines have minor side effects, soreness, a brief fever, as the immune system does its rehearsal. Serious side effects are rare, and monitoring systems watch closely. The benefit, across history, has been dramatic. Diseases that once defined childhood now appear in textbooks rather than hospital wards. The lesson is simple. Our bodies are individual, but our risks are shared. Vaccination, sanitation, and quick responses to outbreaks are the ways we weave safety nets strong enough for everyone. Open your kitchen cupboards, and you're looking at a museum of microbial art. Fermentation is one of humanity's oldest partnerships with microbes. Long before we knew who they were, we learned that certain combinations of time, warmth, and ingredients produced foods that were safer, tastier, and longer lasting. Consider bread. Flour, water, and salt would bake into a dense cracker. Add yeast, weight, and the dough inflates. Yeast eats sugars, release carbon dioxide, and weave bubbles through gluten. Heat sets those bubbles into a tender crumb. In sourdough, wild yeasts and lactic acid bacteria live together in a starter. The bacteria make acids that sharpen flavor and help bread keep. The yeasts lift the dough. Each starter is a little ecosystem, shaped by flour, water, and the baker's hands. Yogurt and cheese are milk transformed. Lactic acid bacteria digest lactose and release acid, which thickens proteins and lowers pH, discouraging spoilage microbes. Different cultures of bacteria, temperatures, and times create different textures and tastes. In cheeses, molds and bacteria ripen curds into everything from creamy brie to sharp cheddar, each a careful choreography of microbes and time. Vegetable ferments like sauerkraut and kimchi begin with salt. Salt draws water out of cabbage or radish, creating a brine. Salt-tolerant lactic acid bacteria prosper, souring the environment and preserving the food. Garlic, ginger, and chili add flavor and small antimicrobial nudges. The result is crunchy, bright, and safe even without refrigeration. In soy ferments, miso, tempeh, soy sauce, molds and bacteria unlock proteins and create deep umami. Cacao beans ferment in their own pulp, driven by yeasts and bacteria that generate heat and precursors of chocolate's aroma. Coffee cherries undergo their own microbial steps that influence flavor. Food microbiology also guards safety. Certain microbes make toxins in warm, anaerobic places. Careful handling, clean tools, correct temperatures, proper acid or salt prevents this. Pasteurization of milk reduces risks from pathogens while preserving nutrition. Canning requires attention to acidity and heat to avoid environments where dangerous spores could wake. At a deeper level, fermented foods are cultural memory. Starters passed down generations contain microbial lineages unique to a family or village. Recipes encode practical microbiology: when to feed a starter, how to burp a fermenting jar, when a curd feels right. Around a table, we eat not just nutrients, but stories of collaboration between people and microbes. The simple truth is delicious. Microbes make our food safer, more varied, and more flavorful. With a little salt, time, and respect, they turn harvests into heritage. We have walked together through the hidden world of microbiology. Step by step, we've uncovered how tiny organisms, invisible to the naked eye, quietly sustain and shape the world we live in. We began by learning what a microbe is, a creature or entity so small that it takes a microscope to see it. At first, we met them simply. Bacteria as single cells, viruses as strange packets of genetic instructions, fungi as recyclers, protozoa as lively swimmers, and algae as plant-like oxygen makers. At its core, that was our starting map. Then we zoomed in. With bacteria, we saw how something so small can be so versatile. They are architects of soil fertility, helpers in digestion, partners in food production, and sometimes, unfortunately, bringers of disease. Their ability to divide quickly, to share genetic tricks with one another, and to adapt under pressure reminded us of their resilience. They are tiny, but they are survivors. We turned next to viruses. We learned how they sit at the edge of life, lifeless alone, but active and potent when inside a host cell. We saw their strategies, attaching, entering, multiplying, leaving. And we recognized how much of human history has been shaped by viral infections. Yet we also glimpsed their role in evolution, in regulating ecosystems, and even in the tools of modern science. We explored fungi, the great decomposers, breaking down fallen wood into soil, partnering with roots in ancient trade agreements, and flavoring our bread, cheese, and wine. Protozoa showed us the liveliness of a single cell, swimming and hunting beneath the surface of a pond. Algae reminded us that every second breath we take likely comes from their oxygen. These groups reminded us that microbes are not one story but many, each with its own role. We then looked inward, to the microbiome. Trillions of microbes live in and on our bodies. They are not intruders. They are residents. They help us digest food, train our immune system, and protect us from invaders. This view changes our perspective. We are not one organism, but many. A walking, breathing ecosystem. We saw pathogens too, the microbes that can cause harm, bacteria that release toxins, viruses that hijack our cells, protozoa that spread through mosquitoes. But alongside the threats, we found defenses, the skin, the immune system, the memory of past battles, and the power of vaccines. We also saw the story of antibiotics, one of the greatest medical discoveries, and one we must use carefully, knowing that microbes evolve quickly. Beyond our bodies, we looked outward again. We discovered microbes as engines of the environment, recycling carbon and nitrogen, building partnerships with plants, influencing climate, and even cleaning up our messes through bioremediation. We saw them not as background noise, but as the very infrastructure of life. We paused to consider how scientists study microbes, the microscopes, the stains, the cultures, the sequencing, all the tools that turn invisible life into visible knowledge. We explored biofilms, where microbes form miniature cities, communicating, sharing, and resisting challenges together. We looked at genetics, where DNA is traded like recipes, mutations spark innovation, and evolution happens at breathtaking speed. We dove into metabolism, microbes breathing oxygen, or sulfur, or iron, or nothing at all. Some using sunlight, others fermenting sugars into acids and gases. We realized that microbes don't just survive in extremes, they thrive. They are chefs of chemistry in every corner of Earth. We touched on immunology, the city guards and detectives of our bodies, and on public health, where the safety of one depends on the choices of many. We saw how simple practices, from vaccines to clean water, save millions of lives quietly every year. And we ended with food. Fermentation is one of the oldest and friendliest partnerships between humans and microbes. Bread rising in an oven, yogurt thickening overnight, cabbage turning to kimchi in a jar. Each is a reminder that when we respect microbes, they gift us with flavor, safety, and culture. All of these lessons bring us to a simple truth. Microbes are the invisible fabric of life. They are not just germs. They are builders, recyclers, partners, teachers, and sometimes adversaries. Without them, there would be no soil, no oxygen, no digestion, no forests, no oceans as we know them, and no us.